one advantage that in the war on ignorance is that in the war of ignorance, unless in other wars, the weapons builders get medals too. So, it is often, and I don't think that in some sense it's a triviality, that it's the methods, the new methods that allow us to go further, and you've seen examples of that today early in the morning, or in the morning in astronomy, where the development of CCDs, of sensitive CCDs, was and large area CCD was a crucial enabling step to discover these objects of the Kuiper belt. Now, there is a similarity between, in particular, fluorescence imaging and astronomy, in that you always have too little resolution and too little light. Now, so if one thinks back about the history of microscopy, then it also shows you how this interplay between understanding and engineering sort of gives us the new methods. So it was in the 19th century that understanding the physics of light enabled various researchers and craftsmen in collaboration then to build microscope objectives that were perfect in some sense. They were no longer limited in their resolution by sort of technical imperfections, but limited by a fundamental quantity of light, the wavelength. Now, that didn't mean that the development of light, of microscopy, stopped at that point, and that physics didn't keep contributing to the development of light, of microscopy. So one example is, and this is the advantage in some sense one has as being a house physicist to neuroscientists, is that that often one can use old physics to create new methods in a different field. And so one could say that multiphoton excitation, nonlinear optics, is old physics, at least theoretically old physics. It has there is an interesting gap, temporal gap between the prediction, which is this paper by Maria Gobbert-Meyer in her PhD thesis in Göttingen, and the first experimental demonstration of multiphoton excitation, which occurred in 1960, shortly after the invention of the laser, because one needed the laser to actually do that experiment. Now, the question is, why would one go through the effort of getting a laser and so on, and effort and expense to coax two photons to actually do the work of one, if single photons are perfectly available to do the excitation that we want to excite, want to drive in a molecule. And so there are, for the spectroscopists, the reasons are that it gives you access to different energy levels, because the selection rules are different for multiphoton excitation. Now, for the microscopists, the selection rule issue is just a nuisance. For the microscopist, the important part is the strong superlinearity of the excitation, and this is demonstrated in this slide, where you can see that you have a really qualitatively different behavior of two-photon excitation. You have really a point of light, and this allows you to build, in some sense, the ideal microscope. Now, if you want to get the ideal microscope, gives you basically information about a single point of space that is not polluted by any information from any other points in space. And even though the confocal microscope sort of does that, it does it at the expense of throwing away a lot of light, because you have to throw away all of this light that's excited outside the focus. And the two-photon microscope allows you to basically build an ideal microscope. which is you basically generate a point of excitation, and then here is the next thing that this allows you to do. It allows you to now be very generous or very undiscriminating in detecting the signal that you generated. It can be fluorescence, as in this case, and so you can use all the scattered fluorescence, for example, to create an image. Now there is an analogy that in electron microscopy, which I will get to, which there's a similar effect. But let's stick to this for the moment. So if you are, and I'll give you sort of approximate numbers, if you have half a millimeter down into the brain, only about 1 in 10,000 photons fluorescent photons will escape unscattered. And so if you can collect those scattered photons, you actually get an improvement of 10,000-fold in signal. And it's rare that one can scoff at such an advantage. And so this is really the reason why two-photon excitation has been so useful in neuroscience. It's this effect. And now, not all nervous tissues scatter. And or one should say there's one nervous tissue that does not scatter, and there's a good reason for it, because in the retina, the image formation, the light that forms the image on the photoreceptors has to go through the processing layers, and if the processing layers themselves were scattering, they would be the image would be blurred. And so by some effects, and I don't think we understand this in detail, the retina is exquisitely non-scattering. It's exquisitely clear. So you can say, okay, so then we can use the confocal microscope to do calcium imaging, for example, in dendrites, which is where some of, as you will see, some of the processing in the retina actually occurs. Now the problem with that is that if you use the confocal microscope, the retina is blind before you can say boo, and or before you can take any data point. You can take exactly one data point, which sometimes is better than none. But if you want to observe something for a while, and you want to observe things like how a dendrite responds to different stimuli moving in different directions, you need to avoid exciting the photoreceptors. And of course, since the two-photon microscope, if you use can use infrared light, and by the time the light has reached, the cone has reached the photoreceptors, it's sort of spread out, and so the two-photon excitation is way down. So we can actually look at dendritic calcium while we're sending moving gratings. And this is something that in collaboration with Peter Detweiler and Thomas Euler, we used to show that the dendrites of starburst amacrine cells are direction selective, unlike the voltage in the soma, which is not. Now, there's other stories I could talk about in measuring activity with a two-photon microscope, but I actually want to switch to what is my current passion, which is, in some sense, which is something befitting, one could say, as one grows older. One works on more static things. One works on structure. Now, structure, if you think about structure, is in neuroscience had a little bit of a bad name, but in some sense, sort of neuroanatomists were not always the hot shots. But if you look at biochemistry, for example, in biochemistry, often the final, or usually the final word on how enzymatic mechanisms work are provided, or is provided. The final word is provided by the atomic level structure. So it's structural biology that, in some ways, reigns supreme in biochemistry. And I think this is why I like to call neuroanatomy sort of structural neurobiology. But that's maybe only semantic. So, and in fact, we've already seen these examples. This is just the same paper that Corey cited, and she made reference to the circuit diagram, to the wiring diagram multiple times. And I think, so there are various reasons why I got sort of personal reasons I got involved in the wiring diagram, but I think it's really, my feeling is that it will provide in more sort of reliable and final answers to to some of the questions, then activity measurements which are very useful to give us clues into what is going on will ever do. Now, there is, of course, you could say this has been done. We have the connectome, we have the wiring diagram of one animal, and everything else is just sort of scaling it up. And in some sense it is true, but the question is sometimes scaling it up is the barrier. And the barrier in structural neurobiology has been that it's been difficult to do other nervous systems. And this is limited both on the data collection side and on the analysis side, as I will point out, I will get into. So now we started out, what turned out to be actually an old idea, which is the serial block phase microscope, scanning electron microscope, which gives us this more or less the same general type of data that serial section electron microscopy in general gives us if you use a transmission electron microscope. But because we are imaging the slice before we cut it off, it's perfectly aligned with the next slice, and the likelihood that we lose it is smaller, and we can cut thinner slices because we don't actually need the material that we remove. So this gets us down to something, slices on the order of 15 nanometers now in the best case, so typically more like 20 or so. But that's much better than say the 40 nanometers that you can do in serial section EM. Now the information that we get from scanning the surface is in fact very similar. And the reason this is a transmission electron micrograph, and this is the block phase that's left behind. The resolution is a little bit worse. And the reason the information is very similar is because in both cases it's heavy metal ions, in fact what matters is the nuclear charge, which scatters electrons in one case out of the beam in the transmission electron microscope and back towards the detector, which is mounted above the sample in the scanning electron microscope. And we just have to invert the scanning image because the signal has opposite polarity. So what have we been doing with that? So we means what's going on here? Something. Okay, so this is Kevin Brigman, who I was very fortunate joined my lab quite a while ago and has now his own lab at NIH. And so he took a retina, filled it with calcium dyes, and then ran stimuli. The little arrow in the corner tells you which direction the stimulus goes. And so then this allowed him to identify direction-selective ganglion cells, and not only identify the fact that they're direction-selective, but also establish their preferred directions. In this case, there are 4 cardinal directions, which has been known before. So then Kevin took this piece of tissue, the very same piece of tissue, and cut it in the serial block phase microscope. Now he had a 3-dimensional electron micrograph, if you want. The data set took about 6 weeks to acquire. And he could start from a cell that actually has a known preferred direction. And so now the next step is, if you want, the most tedious one, where we start out from the cell body and trace all the processes of the cell. So this takes a while, but some of it can be farmed out to undergraduates, and hopefully at some point to the web community, which spends a lot of time idly playing video games, and they could be helping us in neuroscience instead. So we'll see whether we can persuade them by hook or crook. So now, so this is now one bistratified direction-selective ganglion cell. So you will see in a moment where it will hopefully turn. Yeah, you can see the cell body. It's a little bit hard to see the contrast, but you see it's bistratified. And so Kevin then looked at the connections between cells of different preferred direction and with starburst endocrine cells, and he found that there's a strong correlation between the direction of the dendrite of the starburst endocrine cell and the preferred direction of the postsynaptic ganglion cell. Now, one problem in this is that there are really many problems, but one problem in particular is that in this case we had to make a decision So we stain the tissue in a way that emphasizes the surfaces, but this means that we don't have the characteristic synaptic structures anymore, but we have a characteristic shape which allowed Kevin from about 10,000 places where the skeletons came close to identify the true synapses and sort and distinguish them against the mere touches, where the process is merely connected, touches other. And so now this was not what anybody. This is not, I should say, a connectomic project. It's an anatomy project which is very targeted, and it involves quite a number of cells. But so our ambition, of course, was to do connectomics. So we then switched to a data set that also Kevin had acquired, but Moritz now set out to actually analyze this in a way that gave us all the connections between all the cells whose cell bodies we found and that ramify in the inner plexiform layer, which is one of the main processing layers of the retina. So this was now the tracing. Now we needed to scale up the tracing, so we hired a bunch of undergraduates, and they traced all these cells, and they traced them multiple times. So we have a reliability of the structure. Now, of course, they're not exactly the same, but we have mechanisms where we consolidate these skeletons. And so now we have all the skeletons of all the cells. Is about 1,000 cells or so, 980 odd cells that send their process in there. And you can see these blue turquoise layers are the chat bands. There is now a problem, because we don't want to look at all proximities by hand. We don't want to look at all points where these skeletons get close to each other. So we have to somehow get a volume representation. But to get the volume representation takes about 10 times as long, or 100 times as long, almost 100 times as long, than to get their skeleton. So it's impractical and too expensive to do this with human labor. So we need computers to help us. And so here we have some raw data in various cross-sections to the data stack. And so we struck up a collaboration with Sebastian Sung's lab at MIT. Now MIT, of course, has to be mentioned here. So in some sense, I can count myself as somehow having some connection there too. And anyway, so Sebastian, together with Moritz and two of his students, Srini Turaga and Viren Jain, developed a convolutional neural network that serves as a boundary classifier. It basically discovers cell walls or cell membranes. And then we took this and created a segmentation from this. Which largely conforms to the true boundaries. It has a couple of extra breaks. That means there are sort of boundaries between segments that actually shouldn't be there. And so in order to heal these, we now go back to our skeletons and in some ways decorate the skeletons with all the objects. And this is all work still unpublished in detail, particularly. And so now this is the terminal arbor of a bipolar cell axon, and so all of these fragments that have different colors but belong to the same cell because they overlap the same skeleton. Now we basically get now a volume representation. Now the volume representation, of course, is not an end in itself. The point is that if you have now for all cells volume representations, we can now detect contacts between cells, as you can see here in blue. And we can not only detect the contacts, we can also quantify their area. And so from this we can generate a contact matrix, and of course physical contact between cells is a necessary, but not, of course, not sufficient condition that there's a synapse. So this is now the matrix we have. We generated. This has close to 1 million entries. And you can, I sort of labeled some of the landmarks. So the bipolar cells are all on the right side, and they were sort of hand classified, and they conform largely to what one finds in the published literature on cell types. Now, I say largely because we found one cell type that is novel, has been described in monkeys, but not in mouse, really. And we also found something else. Now, if you look at what's called the type 5 bipolar cell, then and you plot it's what's called so plot all its axons on top of each other, then they don't behave properly. Now, what means, how do axons behave properly? In the retina, axonal fields of the same type of bipolar cell should avoid each other. So they should tile, or they should form a mosaic, which these cells clearly don't. Now, if we now look a little bit closer in our matrix and get some help from the connections to two different types, the W3 cell type and some other type of ganglion cell, and only use these cells that preferentially connect to the lower type cell, the ONO1 cell, then we do find a reasonable matrix. So we now have actually used the connectivity in this diagram to classify cells, and this may be a point that. And we have, in the end, in the end, you could say, now we define cells by their neighbors. Good. So now I'm from talking about a project in progress, or that's almost finished. I want to talk about a project that's basically mostly that in the planning stage. Now, and this is we are interested in birdsong, and in birdsong, in one question in particular, there is a theory that the exquisite timing sequence discovered by the Phi-lab, among others, is generated by a Synfire chain. And a Synfire chain would have a very characteristic connection pattern. In fact, it would be, if it's properly sorted, all the connection would be above the diagonal. And so we have hopes that this will yield to a connectomic or a structural neurobiology approach. Now, our big ambition is, of course, to do a whole mouse brain. And Sean Mikula is working on this project, and the first thing we need to do is actually stain a whole brain for electron microscopy, which we can do basically by soaking it long enough. I mean, there's a few other tricks, so it's not quite as simple, but because you have to fight diffusion, and diffusion is a formidable enemy if you have to overcome large distances. And so this we now have a brain, a preparation method that allows us to stain a whole brain. Now here you can see a cross-section of this whole brain. It's stained reasonably uniformly. And if we zoom in, now this brain is not stained. Our goal here is to stain myelination well, because our first project is to try to get sort of a myelin projectome rather than getting a complete connectome. And because the complete connectome would just take too long, and I'll get to that in a second. So this preparation is well stained and can be cut, as the little movie here shows. These are myelinated axons that they could easily follow with your eyes, and it's probably not inconceivable that you could follow them with the computer also pretty easily. Now the problem is, so this we estimate if we can get a 40 megahertz scanning rate, then we can do this in roughly a year at 80 nanometers resolution, which should give us the myelin connectome. Now, if you want to do this at 20 nanometers, which would be necessary to do the cellular connectome and really follow all processes, then it would take us about 50 years. Now, that's not practical for many reasons. So, fortunately, there is a technology development that has come out of Zeiss recently, and where they actually have a electron microscope that has 60-odd beams, maybe it's more in the future, and so it has a aggregate data rate of a gigahertz or more, and so that could push down the acquisition time to about to below a year. And then, then we have a lot of hard disks full of data. And then in some ways you could say the problem begins, because how are we going to analyze this data? Now, if we could get only a small fraction of the time that people, even only in, say, all of Europe or all of America, spend on video games every year, we could probably do this pretty quickly. So now that is maybe not realistic, but having a data set like that will, I think, spur on the computer scientists to really say, here's a worthwhile goal, and they may actually do it for us or help us enough so the leverage is large enough so we can get we can do this with a reasonable amount of human effort. Okay, with this I'm done. I would like to thank the Kavli Foundation, all the organizers, and so on for putting this together, and of course for the all I have to do is roll the credits. I've mentioned most of the people that that were involved. I should say this wouldn't have happened on this without inspiration from a few people that I encountered during my wanderings around neuroscience. And I mentioned Kevin. I think I should mention Heinz Horstmann, who has been instrumental, a very gifted EM technician, then our shops at Max Planck, our electronics and software people. I mentioned Kevin and Thomas here again. I think everybody else was acknowledged already. So thank you.